What is the title on the YouTube?
Has not yet started. Maybe the participant name might have been appearing on that. Now it will start. Good afternoon, participants. I welcome one and all to the afternoon session of Science Academy's lecture workshop. I welcome Dr. Deepak Modi sir to give his lecture on modern techniques, that is clustered, regularly interspaced short palindromic repeats. Over to you, Modi sir.
Very good afternoon to one and all. Thank you very much for having me over again and an opportunity to speak to you. I hope you have enjoyed the pre-lunch session, and this is one of the toughest sessions to take over because post-lunch it's very tough to keep the audience awake, and it's much harder to even talk after listening to some of the real heavy-duty talks, but I'll try my best to make it as easy and as entertaining as possible. There is some video that I have to share during the presentation. I'm not sure how to do it. So maybe in between we might have a little bit of interruption. One of our staff who is handling this event would be coming and helping me out. But without much ado, let me start the presentation. Hopefully he will be here in a minute or so, and he should be able to sort it out. Everybody can see my screen, please. Just a minute, please.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA.
DNA consists of two strands twisted into a
A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species.
Is this video audible?
Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not.
Dr. Haleem, is this video audible?
Has been developed that promises to dramatically improve our ability
very clear, sir.
Any species.
Okay.
Audio and video both are very clear, sir.
All right. Okay, thank you.
The CRISPR
based on a natural system used by bacteria to protect themselves.
So I have to play and then start. Yes. Okay, thanks. Sorry.
It's okay, sir.
Okay, so I'll start my presentation, which, as the title says, it is on CRISPR. And when we use the word CRISPR, we all think of chips, we all think of wafers, and we always think that CRISPR the better. And same thing applies to this CRISPR as well. It's one of the better known technologies and better work technologies, and one of the best innovations that's happened in the last decade or so. Just a minute. This year, I mean, all of you have heard about the Nobel Prizes, and this year's Nobel Prize has been very peculiar and very particular and very exciting for several reasons. One of it is that it has increased the number of women who have shared this prize. Imagine till last year, that is 2019, more than 840 men have received the Nobel Prize, and only 53 women have received the Nobel Prize. So there is a huge discrepancy between the number of people who get the Nobel Prize, especially there's a gender bias. But this year, 2 women got a Nobel Prize in chemistry. which is unusual. Not unusual, but it is not usual as well, because you see that there is disproportionate number of men who get a Nobel Prize over women. And these two women have made one very, very remarkable discovery, which comes from very basic research, and it has gone to a lot of clinical applications in basic science, and it is even seen that it has treated people from life-threatening diseases. And this discovery was about the CRISPR technology. It was Emmanuel Carpenteri and Jennifer Doudna who received this prize. Emmanuel is from the Max Planck Institute, the Institute of Pathogen Infectious Biology, and Jennifer is from University of California, Berkeley. And they discovered this technology of CRISPR. So what is CRISPR? How does it work? What does it do? How is it useful? And because this particular seminar is more focused towards COVID-19, I'll very briefly touch upon how CRISPR has been useful in diagnosis and treatment of COVID-19, or what are the perspectives of CRISPR in understanding of COVID-19. So before I start on CRISPR, I will run you through very, very basic biology. My apologies to all those who know this, but I think it's very important for the students here to recall this, that we have about 7 trillion cells in the body, 70 trillion cells in the body, and every cell contains a nucleus. The nucleus contains the DNA. The DNA is wrapped up onto chromosomes, and these chromosomes are the functional units or the structural units of the cell. And it is this DNA in every cell of the body which tells us what we are today. So the DNA is wound up around the histones. The histones form the chromatin, and the chromatin then form the chromosomes. But let's focus on this DNA, which you all know that it is a functional unit of the genome, and the DNA is organized into genes. So we have long stretch of DNA, which is made up of just 4 nucleotides, A, T, G, and C, in various permutations and combinations. And this entire stretch is called as the DNA. In the DNA, segments are there. Segments of DNA are called as genes. What are genes? Genes are the units of the DNA which make protein. So every gene makes one and only one unique kind of a protein, and each of this protein would have a unique function. For example, the lactase gene would make the lactase protein, which is required for sugar breakdown. The myosin gene is going to make the myosin protein, which is required for mechanical support for the muscles. The collagen gene is going to make the extracellular matrix proteins, and the globin gene is going to make the hemoglobin, which is required for blood transport. So on and so forth. We have nearly 40 to 50,000 numbers of genes in the human genome, around same numbers in the mouse, and the different organisms have different numbers of genes. Now, these genes have to make proteins. So this you all know, transcription and translation, but just to recapitulate, that the DNA is faithfully copied by a process of transcription into a RNA. This RNA goes to the cytoplasm, and in the cytoplasm, the ribosomes read this RNA to form the polypeptide, which is eventually going to form the protein. And this is the triplet codon, which was discovered by an Indian, Professor Khurana. We know about him. And he said, and it is known, and he has discovered it, that 3 letters is equivalent to 1 amino acid. And the sequence of these 3 letters is very, very crucial. For example, AUG would be read for methionine. When it is AUC, you would add isoleucine. When you have UCG, you will add serine, and so on and so forth. So this is how the DNA is translated to RNA. RNA is translated to protein. Now imagine a situation where this AUG and AUC. This AUG would mean a DNA TAC and a TAG. Now just imagine if this G gets replaced by a C. So now, instead of a sequence which should be reading as AUG, AUC, it is going to be read again as AUG, AUG, and instead of the isoleucine, you would be replacing it by a methionine. Now, when the protein sequence change, that means when the amino acid sequence change, that means a completely different protein, and this would mean a different function or gain of function or loss of function of the protein. This I'll explain you slightly more simple way that you can have two kinds of genetic changes. One are called as single nucleotide polymorphisms or SNPs. These are silent changes which do not have major function on protein functions, but it doesn't mean it's really not of any use. For example, I spell my name as DEEPAK, or I can spell my name as DIPAK. So you can see this double E is replaced by an I. Now this means that it doesn't really matter under most circumstances whether somebody writes my name or I write my name as Deepak or DIPAK Deepak. It would essentially mean the same. Of course, legally it might have some issues, but in general, DEE versus DIPAK would not make any difference. And this kind of a change in the DNA would be called as a single nucleotide polymorphism. But these polymorphisms are very crucial because it differentiates all of us from each other in terms of our skin color, hair texture, our behavior, and many things. Nevertheless, there are some changes which are mutations. What are mutations? These are changes in the DNA sequence that affect protein functions or protein production. For example, I have to write my name as DEEPAK to be spelled as Deepak. Now, suppose something happens and the K is dropped off, and from Deepak it becomes Deepa. My gender changes. If somebody calls Deepa, I will not respond. So that means that such a change in my name sequence is a mutation. So any sequence in DNA, which would mean a change in protein sequence to an extent that the protein function is lost or the protein function is changed, is called as a mutation. And these single nucleotide polymorphisms, mainly mutations, are responsible for many genetic diseases. Because, as I said, that a small change, even one nucleotide change in the DNA sequence can lead to difference in protein function or protein structure. For example, here you see one DNA sequence variation and the protein folds in a different way. And if the variation is just switched from G to A, this folding pocket, the folding of this protein changes very significantly. And this would mean many things. It might not interact with other proteins because now the shape is different, so it cannot interlock with its receptor, or its shape is different, it cannot partner with another protein, or its shape is different and it cannot now attach with a substrate, and the enzyme kinetics might get altered. So such changes are often detrimental because these small differences can determine whether we are tall or short, hair color, our hair texture, our skin color, eye color, so on and so forth. But these differences are sufficient to make some people very healthy and some people very sick. It can determine whether we can get cancer or not. For example, BRCA gene mutations, it can predispose us to cancer. A beta globin gene mutation can give us thalassemia. And women who, for example, as already told you, that women who carry a genetic variation of the BRCA1 gene are 7 times more likely to get breast cancer than the women who don't carry. That means these genetic changes are one which predisposes us to diseases. And if we are missing certain nucleotides in the specific spots on the genome, we might get things like cystic fibrosis. So the larger question that scientists has always been, now genetic diseases for these many years had been thought that these are not correctable diseases. We cannot correct genetic diseases because we cannot edit the genome. But for years since the discovery of the genetic diseases, scientists have been trying very hard to figure out if these genetic diseases can be corrected, because when an individual is already born, there's no treatment for genetic diseases except for correcting the mutation. And this mutation correction is now possible with the help of genome editing technologies like CRISPR. Basically, what is CRISPR? CRISPR helps, it changes the DNA sequence, or it can help us to remove a DNA. So they're like molecular scissors, which can help us to remove the DNA sequence and allow it to paste a new sequence in its place so that the defective sequence can be removed and a good sequence can be added. So, for example, This CRISPR technology, as Sir has already said, stands for Clustered Regulatory Interspace Short Palindromic Repeats. Now, what is this? I'll show it in a video. I'm not going to speak much about that right now, but it requires a protein called as a Cas9, and it requires a guide RNA and a different RNA strand. This, when loaded together, can go inside our nucleus and scan for homologies with this region. And once it finds a homology, this Cas9 protein can get activated. It can cut the DNA at the right places, and it can replace with a new sequence or repair the original sequence. So the machinery that you require for CRISPR, as I said, is this wonderful protein called as Cas9. Earlier, Cas9 was the protein, but now there are multiple Cas proteins identified. These were first identified in the bacteria, and these proteins these proteins have been engineered significantly, and now these have been one of the most powerful tools that have been discovered by the two women who received this Nobel Prize. So of course, Cas9 proteins were known in the bacteria, but this Cas9 protein load onto another component called as a CRISPR, the guide RNA component, which has a target specific CRISPR sequence and the TRAC-CR RNA, which is required for functioning of the Cas9 protein. So what essentially happens is that the Cas9 protein loads onto this CRISPR RNA. It is going to go onto the DNA, scan the DNA, and identify regions of homology. Once it identifies regions of homology, the Cas9 protein cleaves this region. That means it creates nicks in the region, and it can leave the DNA, and it can just repair by non-homologous end joining NHEJ, and the region can be deleted. So suppose if somebody has extra nucleotides, by simply designing a guide RNA for that region, you can just remove those extra nucleotides, creating a deletion called as indels. Alternately, suppose if somebody is missing a nucleotide or we want to replace a nucleotide, you can do something like a copy and a paste. So you can have a guide RNA, which you want the right kind of a sequence. It should go to the regions in the DNA. It will just cut. It will now not just cut, but also paste it in that region by homology directed repair. And now you can have completely new sequence inserted into the genome of the individual. So I'll share a video here. I'll stop sharing the screen. I'll share this video, which is one of those wonderful ones which I've come across on CRISPR-Cas9. We will try and see what we can. I hope I'm able to play it now. Is this visible to everyone on full screen now?
Yes. It has to start. It's visible.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
So I hope the principle of CRISPR was clear to everyone. So this is how CRISPR functions, and this is what the Nobel Prize was given for. But remember, the Nobel Prizes are not just given because somebody has made a discovery, but it is thought that it has a more widespread application. And as of today, CRISPR has found tremendous applications in gene surgery, because when we have mutations, we can correct them through CRISPR. I'll give you 6 examples for that. It has been very useful for drug development, developing animal models for research, genetic engineering, creating various variations so that new cell types can be made. It is useful for making materials, it's useful for making foods, fuels, and also combat bioterrorism. So what is the vision of CRISPR and how it has been very useful? So one of the early trials of CRISPR was in patients with beta thalassemia. Beta thalassemia is a disease in which the patients don't make enough hemoglobin because there's a mutation in the beta globin gene. What researchers have done is they've collected the bone marrow from these patients with beta thalassemia. The stem cells from the bone marrow were isolated. In these stem cells, genome editing was done such that the mutation was repaired, or a block which reduces the synthesis of globin gene was removed, and there was increased expression of fetal hemoglobin, and such cells were infused back to the patient, such that now the patient is able to make enough hemoglobin, although of fetal type. It was able to rescue the patients from transfusions, for not having continuous transfusions. And this modified stem cells were found to be of high therapeutic efficacy. So simply by editing the genomes of the bone marrow stem cells of the patient, one could actually result in a treatment of a condition which was previously never been possible to treat completely. CRISPR has been thought to be extremely useful, or people are trying to apply CRISPR for HIV therapeutics. What we know is that HIV is a integrating kind of a virus which integrates into the genome of individuals, especially in the CD4 plus cells. And in these cells, along with the replication of the cells, the HIV genome also replicates. And while this replication is happening, the virus is made, and the virus then eventually lands up killing all the immune cells, and the HIV-infected person moves to AIDS. However, if we delete this virus from the genome somehow, the virus production can be hampered, and then therefore the person can be cured of HIV. So the vision here in HIV therapeutics is that if we can design a specific CRISPR system where you have the guide RNA and the right Cas9 to go and target the HIV genome, one can remove the important sequences of the HIV which is required for the multiplication and integration, and you would have T cells without any HIV genome, or only non-functional remnants of the HIV genome remain behind, and you can and the person can be free of HIV infection. But beyond this, one of the very interesting ways that people have actually landed up curing HIV using CRISPR is by deleting CCR5. Now we know that HIV infection requires a CD4 receptor, but also requires a co-receptor on the T cells, which is called as a CCR5 receptor. So HIV primarily binds to the T cell and then gets activated by CCR5, and only then it can get inside the cells and eventually killing the T cell. What we knew from literature was that there are some individuals who are resistant to HIV infection because they don't have CCR5 gene. There were naturally occurring mutation called as delta 32 mutation of the CCR5 gene, which is making them resistant to HIV infection. And these individuals were happy, healthy, hearty. They had no other diseases. That means removing 32 base pair from CCR5 does not do any harm to the individual, but in fact, it is protective against HIV. So what people thought was that if there's somebody infected with HIV, if we can delete the CCR5 gene deliberately, so here now you are actually creating a new sequence, you are actually removing sequences from the individual, creating a new mutation in the individual such that the patient becomes resistant to a disease. And what was done was that they developed a guide RNA specific for CCR5 locus, matched it with a Cas9 protein, and then they were put in the T cells, and the T cells were engineered such that they don't no more make the CCR5. And the idea was to make these cells resistant of CD4. There was this group in 2014 that published this work. They took again the bone marrow stem cells, which are going to make the T cells. They took the bone marrow stem cells from these individuals who were infected with HIV, and they did genome editing with CCR5 in autologous CD4 T cells. They infused back the T cells into the same individual, and then they looked at CD4 T cell, whether these CCR5 negative CD4 T cells were remaining in circulation, and indeed they found that these cells could survive long enough in a HIV infected person. This study was published, I think I missed writing the date. The study was published late last year. Very, very interesting. They CRISPR edited the stem cells of the patient with HIV, and he also had acute lymphoblastic leukemia. Because of his HIV status, they deleted the CCR5 locus from the individual, and they infused back the stem cells by a bone marrow transplantation. And look at the data. This person was on antiretroviral therapy, so his viral load was very, very low. After infusing the cells in the body, the antiretroviral therapy was interrupted for quite some time. So when the interruption occurred, they did this interruption for around 20 or 30 days. No, I think they did it around 50 days. And they found that as soon as the antiretroviral therapy was interrupted, the viral load started accumulating and it increased. But eventually this increase, otherwise if the patient was not on drug, this patient would have increased further. But because his T cells were engineered, so the CCR5 would not get in, and they actually found that the viral load decreased and it became to ground zero. And this patient was in complete remission. In spite, he was not taking any antiretroviral therapy. Look at his CD4 counts. The CD4 counts, normally CD4 counts decline upon HIV infection, and especially the HIV increases, the viral load increases, there's a drastic decline in CD4 count, which they observed for some time, but the CD4 count recovered to that of the pre-treatment level. I mean, at the treatment levels, and the CD4 count could be maintained. And this is CCR5 modified cells in the blood. They found that as soon as the retroviral therapy was interrupted, the CD4, the CCR5 negative cells peaked and the levels came back. That means that the virus was released off from the existing T cells, but it could not infect the new cells, and the patient eventually removed all the virus because the new cells could not get new T cells, could not get a new host, and the virus could be completely cleared from the system. So look at the power of CRISPR here. Simply by removing some nucleotides of the CCR5 gene, somebody who is infected could be cured of HIV, and somebody could be even protected from HIV. And this work was in fact done in China, although it has got into a lot of ethical debates. They took human embryos and they edited from a couple who had HIV, and the couple was terribly worried that what if their baby also gets HIV during the course of the pregnancy. And what this researcher, Professor He, did was that he edited the genome of the embryos to remove the CCR5 gene, and two babies have been born called as Lulu and Nana, who are now born in this world with the genome edited even before at the time of fertilization itself, and now they would be truly resistant to HIV for life. So this is the power of CRISPR. Now, in this case, they use a CCR5, but one can use beta thalassemia, one can check for BRCA mutations. Name any disease where a genomic correction has to be done, and CRISPR comes in very, very handy because it's very easy to use, very robust, very reproducible, and not at all expensive, unlike gene therapies, which are quite expensive. And not just in case of HIV, but clinical trials, therapeutic trials for CRISPR have been ongoing. So there are various ways of delivering this CRISPR complex into the cells. You can do by adenoviral vector deliveries, you can do by non-viral delivery, and you can even do a physical delivery, like in case of embryos. This has been done. And for neurological disorders like Parkinson's, Alzheimer's, Rett syndrome, autism, CRISPR therapy is currently ongoing. For many monogenic disorders, of course, CRISPR is one of the best ways to treat these monogenic disorders like Duchenne muscular dystrophy, cystic fibrosis, beta thalassemia, hemophilia, Huntington disease, so on and so forth. Even non-monogenic diseases like Fanconi's anemia and cancers, which are breast cancer, for example, BRCA mutation. If you can correct the BRCA mutation, the breast cancer can be regressed very rapidly because the cancer cell cannot now proliferate. And this has also been in clinical trials. Now, for several viral infections, HIV I've already told you about, but like HIV, hepatitis, HPV, HCV, EBV infections, these are all very deadly infections of the virus, and CRISPR has come in very handy. And of course, for ocular diseases like the cataracts and retinitis pigmentosa, CRISPR-Cas9 clinical trials are ongoing. Well, now very briefly touch upon how CRISPR is of help in COVID-19. CRISPR, we could think of how CRISPR could be useful for treat COVID-19, but in fact, the application of CRISPR has actually come into diagnostics of COVID-19. This is something that Professor Dodna had proposed, and one of the companies which she floated has initiated this, and this diagnostic technique is already in the market. What they have done is they have developed a Cas9 and a CRISPR. So now you can see from Cas9, now they've come to Cas12a and Cas13a, which are genomically engineered Cas proteins, which will help in better functioning of the CRISPR system. They have developed a CRISPR molecule which has fluorescently labeled probes And these probes can go and bind to these probes have homology to the COVID-19 genome, the SARS-CoV-2 genome. And now what you simply do is take the salivary preparations or swabs or blood, semen, whatever that you would want to think you take, and incubate it with the CRISPR molecule. And this CRISPR, as I said, have reporter molecules which are intact, has one side a silencer, a quencher, and other side a fluorescence. And you can incubate it with the CRISPR DNA and either run a gel electrophoresis. So if there is a viral protein there, the presence of the viral protein, if there is a virus RNA there, the presence of the virus RNA is going to allow this probe to get cleaved because of the CRISPR, and the cleavage product can be visualized by agarose gel electrophoresis or lateral flow strips. Lateral flow strips are nothing but these small strips on which the pregnancy kind of tests are being done. Similar strips have been developed where you can track the migration of this, and you can know that if there is presence of the virus, this probe will get cleaved and the probe will then move at a differing motility, and one knows there is presence or absence of the DNA. Or you can simply quantify the amount of the target by simply quantifying the amount of probe released because of the cleavage, and increase in this fluorescence intensity would mean there is presence of the virus. This has been very, very rapid. It is so remember currently when we apply the PCR technology, which requires to do RNA extraction, which requires to do PCR amplification and quantification, which in the shortest period of time at least requires, the PCR itself requires 2 hours. It requires at least 30 minutes for the RT, another 30 minutes for setup and RNA extraction, and so on and so forth. This means that a typical result would come in anything about 6 to 8 hours. And remember, it's not a point of care because you need to collect it, send it to a specialized laboratory. Now, this CRISPR technology has been made so user-friendly that you can put it in a simple solution, and the solution has to be just mixed with the extracted RNA, and on a fluorescence reader or on a lateral flow strip, you can read it. And this entire CRISPR-based genomic detection of the SARS-CoV-2 is possible even on a point of care. So today our point of care are the rapid antibody tests or the rapid antigen test, which has its own sets of issues. But because this does not involve the use of PCR, it is faster, it is much more economic, and remember that it is possibly going to be the next point of care test going to be available for COVID-19. So this is how CRISPR technology from therapeutics has actually first came into useful in COVID-19 diagnostics, and the company has already patented this, and it's now going to be available in the market once the adequate regulatory approvals have been obtained. Of course, for the virus, the vision is to make the vision is to develop CRISPR against the viral genome and give it inside the cells, and any cells that have the virus, the CRISPR should get activated and actually cleave off the viral genome, like how the bacteria do. When the bacteria gets infected with the bacteriophage, they activate their endogenous CRISPR system and cleave the viral RNA. The idea here is that human cells should have the CRISPR system and the necessary machinery against the SARS-CoV-2 virus, and the virus can be simply cleaved because of the presence of the Cas9. So various kinds of Cas proteins are being tried, and various kinds of guide RNA are being tried to inactivate the SARS-CoV-2 such that it could have a therapeutic potential. This is, of course, in a visionary stage. People are still yet to develop the right kind of probes which is required to cleave the Cas9, CRISPR Cas9 RNA. The SARS-CoV-2 RNA. The challenge here is that now your guide RNA should not target any human gene. If there is any sequence homology, it's going to go and kill the actual human genomic DNA, and instead of killing the virus, we might just introduce mutations in our cells. So this is one of the major limitations as of today the CRISPR technology is facing, is to design the guide RNA. But in time to come, we should have the therapeutics for COVID-19 using the CRISPR technology. the CRISPR technology in the past was facing a lot of issues with off-target effects of guide RNA. It would target at the right place, it would go even at the other places. The Cas9 was not very efficient in working in the human genome, but of course, in the last 5 years, large numbers of technologies have evolved, which has improved the Cas9 and the ability to make correct guide RNA, and it is constantly improving, but the off-target is not yet zero. There are some off-target effects, unwanted effects of the guide RNA. The major challenge is delivery of the Cas9 protein loaded with the guide RNA. It is easy to put this in a test tube, but delivering it inside the cells in a lab dish is reasonable to do, but it is very, very hard to deliver these cells in the body. You cannot just pop a pill and it just goes there. How to deliver these cells into the cells of the throat? For example, if you want to kill SARS-CoV-2 in the lung, it's very difficult to deliver these proteins because, as I said, that the virus, which is much smaller, cannot get inside the cells. The Cas9 protein has to be targeted to these cells. A challenging task that people are trying to figure out. And nanotechnology is thought to be something which is going to come to rescue for the right kind of delivery of the Cas proteins at the guide RNA. Another challenge is inactivation of the Cas9 complex. Many a times when this Cas9 complex gets inside the cell because of the cell physiology, the Cas9 complex gets inactivated and it cannot function. And success rates are not always 100%. All the examples that I've shown you are merely case reports where 1 or 2 people after a lot of exercise have been able to treat. So it's not that the treatment of Cas9, CRISPR-Cas9 is here in the market tomorrow, but we can see the light at the end of the tunnel. I think in years to come, we should be able to treat many, many genetic disorders thanks to this wonderful technology. And the future is immense. There's a lot of basic research yet to be done to understand the cell and cell function. And CRISPR is going to have tremendous applications. In our lab, we are planning to use CRISPR now to edit certain genes in the cells so that we can understand how pregnancy initiation and pregnancy failures can occur. In medicine, I've already alluded you how important is CRISPR and how it's been useful to even treat things like thalassemia and HIV infections. And in agriculture also, people are thinking of editing plant genomes such that they can make better crops, drug-resistant, infection-resistant crops, parasite-resistant crops, such that we can have good quality crops which can survive in drought conditions. So we know those genes, but now if we are able to edit them endogenously, it should be wonderful to be useful in agriculture. And I'll stop here and I'll be happy to take questions. Thank you once again for having me.
Thank you so much, sir, for your wonderful talk on CRISPR. That is clustered, regularly interspaced, short palindromic repeats. I request Dr. Mahendra to quickly give response on his observation in the lecture. Dr. Mahendra.
Oh, it is wonderful technique.
Madam, you are muted.
Dr. Mahendra, are you available?
So actually, it's a very good technique and it's going to be useful later on also. Of course, therapeutics right now it's not possible, but at least one can detect it. That is what is more important. Okay, only you said it's not 100% good. So it's not compared to RT-PCR, it may not be a very good technique. So RT-PCR is the gold standard right now. So this is maybe they will improve and then it can be useful for detection. Therapeutic it will take a long time, it will not be immediate, but it's very good technique and I think especially for thalassemia and other cases, this will be really useful because people are suffering with, number of people are suffering with thalassemia, so if that can be corrected, it will be wonderful. Thank you. Thank you very much for giving in details all the technique and information about it. Thank you.
Dr. Mahendra, over to you.
Yeah, thank you. Thank you. Good afternoon. It seems the seminar is going on in a good way. I congratulate Haleem Khan and all the team of the department. I'm very delighted to have a latest technique, a CRISPR-Cas technology, which is revolutionizing the total, science, I can say, because it has got a lot of implications in every area of science, I can say, starting from plants onwards, even animals, and it is also influencing the medicine. It has a lot of implications in vaccine production, in corona vaccine production. Thank you very much for enlightening us, sir. Thank you very much. It's a very good and very lucid, to say, explanation, and these basics are must for students to have a lot of to develop a lot of concepts and to make use of this technology in coming days for the human welfare and for the environment. Thank you, sir. Thank you very much. Thank you.
Now I request Dr. Aruna ma'am to speak, to introduce our next speaker, Dr. Akhil C. Banerjee, Director, Institute of Advanced Virology, Kerala. Over to you, Aruna ma'am.
Good afternoon to all the members who are present today in the workshop. And now it's my turn to introduce an eminent speaker, Dr. Akhil C. Banerjee, sir, who is going to deliver his talk on multiple vaccine approaches against COVID-19. Akhil C. Banerjee, sir, is serving currently as Director, Institute of Advanced Virology at Kerala, and has been Emeritus Professor, Department of National Institute of Immunology, New Delhi, and he's also recipient of Emeritus Professorship of INSA and ICMR, that is Indian National Science Academy and Indian Council of Medical Research, and is member of National Academy of Sciences and Fellow of Indian National Science Academy. And Thomas Reuters recognized him as a distinguished expert and leader in virology in 2011. And he has published more than 110 papers in international peer-reviewed journals in the field of HIV, biochemistry, and gene therapy. And he is conferred with many awards and honors, and few of the important ones are: he is recipient of National Awards of ICMR, Shakuntala Amirchan, 1982, National Foreign Associateship Award of DBT, 1998, National Bioscience Award for Career Development of DBT, 2001, SR Scientist Indian Immunology Society Oration Award, 2012 of Government of India, Ranbaxy, Award for Medical Research, 2014, and Lifetime Achievement Award for HIV/AIDS Research. And he also won several full international scholarships to present his work on HIV in USA, UK, South Africa, UK, Japan, and Singapore. And he has guided more than 12 PhD students and trained more than 24 students and postdocs. And he is also recipient of Indo-US NH ICMR grant on HIV transmission and several DBT, CSIR, and ICMR extramural grants on HIV pathogenesis. He's member editorial board of several international journals. He regularly reviews papers for antiviral research, gene, scientific reports, biochemical, virology journals on regular basis. And he has delivered invited seminars in Albert Einstein, New York, Duke, NC, UCLA, CA, COHCA, NCI, NIH, and Drexel, Philadelphia. And he was awarded the visiting professorship at Colorado State University, Frost Collins, Colorado, USA, for a period of 2 years during 2002 to 2004. The work carried out on siRNA, that is small transferase RNA into stem cells against HIV, was covered in New York Times in 2003 and now in clinical trials in USA. His major interest now is to use lentiviral vectors, stem cells, catalytic nucleic acids, and small interfering RNA, that is miRNA technology, as a gene therapy approach against HIV and other pathogens on one hand, and determine the functional implications of genetic changes in the host genes or viral genes in order to understand the molecular basis of HIV and AIDS pathogenesis in India. The project is dealing with microbiology, epidemiology, biochemistry, virus infections, epidemiology and diagnosis, gene therapy, cell biology, life sciences, stem cell biology, development biology, and microRNA technology in plants and animals, biotechnology, national programs on immunizations, vaccine technologies have been evaluated at national and international levels. And with this brief introduction, I now welcome and invite Akhil C. Banerjee sir to deliver his talk, and thank you for giving me this opportunity. Over to you, sir. Thank you.
Hello, am I audible?
Sir, you're audible.
Am I audible?
Yes,
You're
yes,
audible
yes,
and visible, both, sir.
Okay, okay. Good afternoon, and many thanks to the organizers who have given me this opportunity to talk something about the vaccine platforms. But I would like to make it more general virology type of lecture, and with the sole purpose that there are some interesting features about this virus, although I must confess we are still learning. And there are many secrets of this virus yet to be known. Okay, so let us be humble and accept the fact that there are many things that we need to learn from this virus. Okay, can I have the first slide? Share screen. Huh? Okay, is it? Yes. Okay. Yeah. Can the slide, people can see the slide?
No, sir, your screen is not yet shared.
Okay, okay. Please bear with me. I'm taking help from one of the very efficient students.
Will close the rejection in the system.
Oh, yeah.
It will generate all those PDFs of
Yeah.
Sir, now it is visible.
Now it's visible. Okay, great.
So, sir, are you comfortable with the mask?
Okay, I will remove it. Don't worry.
It's up to you. I leave it to you. If you are comfortable, no problem. We can hear you.
Okay, okay. Then let's. Okay. Okay. So I have joined this new institute, and this is known as Institute of Advanced Virology in Kerala. Purpose of this institute is to do high-end research for the benefit of the society. And certainly at this moment, COVID virus is our will be main focus. Although we are only 2 faculty at this moment, but we will soon appoint more faculty members. Okay. So next slide. Okay. So this is a brief history of this institute. Those who do not know, it is very, very new, and this is essentially we will be focusing on viral emerging and re-emerging viral diseases. And this is the various departments that we will have. Okay. So all this information is available on the website of this institute, so I will not dwell into any details. Okay. You may, those who are interested can easily go into the, okay. So Now, let us focus, although it has been, although I have not attended, I must confess, all the lectures this morning, but this is typically of an RNA virus. So this is, of course, more designed towards COVID, but many viruses would fit in this. For example, influenza can easily fit in with this structure, okay? Influenza, here, let us say, this is especially for students. This is, can I have the cursor? Correct. Okay, so this is the spike protein, okay? In case, why am I focusing more on spike protein? Because this is the protein on the surface of this membranous virus, okay, let's say COVID, okay, against which most of the immune responses are made in the body. So if you will ask me a question, see, all RNA viruses, they are prone to changes, okay? That means the faithful copy of the RNA is not made, okay? Am I still audible to the audience? Hello? Am I audible?
Sir, you're audible, very clear, sir. Your sound is very clear.
So the point that I'm trying to make is all RNA viruses, they are prone to changes, but some change more, some change less. For example, if you take, see, HIV also will very fit in with this structure, okay? So here, if this was HIV, just forget about this blue part. This will be GP160 in HIV, okay? And it is this envelope protein, okay, against which most of the antibody responses are made, okay? And not all responses are protective, okay? So some are protective, some are not protective, okay? So this is the challenge. In the hemagglutinin gene, okay? This is where you will find most of the changes, okay? So every year you have to change or use the hemagglutinin circulating at that time. Influenza has an additional problems that it can, as I said, reassort. What do I mean by reassort? Let us say a virus that is circulating is of H1N1 type, and then at the same time, you can have H2N2, so they can reassort and you can have H1N2, or let's say H2N1, okay? So this creates a problem, okay? So this is a problem with RNA viruses. What is the reason for this change problem? That the enzyme that is present inside the virus particle, okay, is defective. Defective in the sense it does not faithfully copy another RNA. Okay, so multiple copies have to be made, okay, inside the body, but it does not make. So we end up having small changes, okay, in the RNA genome, okay, and this is what we call quasi-species. Quasi-species. So let us say a person has been infected with HIV day one, and let's say a typical course, now of course with drugs things have changed. When the drugs were not there, the person will take 14 to 20 years to develop the disease. And if you sequence, keep sequencing what virus changes have taken place over the years, then you will see most of the changes, very dramatic changes, have taken place in the envelope gene. It changes, changes, and changes. Body makes an immune response, okay? Virus escapes that immune response. Again, the virus, again, the body makes an, so you continuously make escape mutants. Okay. It's quite possible that the spike protein of COVID can also change, and we know it changes. Okay, there are some very known mutations that have been found. Okay, and I'm trying to find where did I write it. Okay, it is something with the aspartic to proline. Okay, in the receptor binding domain. Okay, and that seems to be set, and that's in the spike protein, and that seems to be helping with the extraordinary power of transmission. See how this virus gets transmitted so powerfully from one person to the other. Okay, so this is one research question that we would like to address. Okay, here is it. The mutation is called D614G, aspartic to glycine, I think. Okay. One mutation. There are other mutations also, but the mutations that count more are the one that are in the spike protein. There is an additional problem with spike proteins, okay? And so I think not so much with COVID, but let us say with HIV envelope protein. The additional problem is that it is very, very heavily glycosylated. Students, watch my words. If a protein, based on your size, you can predict and run it on a gel, let us say you find that, okay, it will run as 60 kilodalton. But HIV envelope protein at the same time will run as 120 because there are so many glycosylation sites. It is mind-boggling how many glycosylation sites are there. Okay. What is the fallout? The fallout is that most of the epitopes get protected, that gets hidden because of this, almost you can say a jacket of carbohydrates that this protein is wearing. Okay, so this is additional problem. Okay, then you can make neutralizing antibodies. In case of HIV, not much information about neutralizing antibodies, although there are very good studies, sort of people are doing it. Not so much as we know about HIV. The neutralizing antibodies, it will take time. Okay. They don't, if a person gets infected with COVID, that person will take 2 to 3 months to make neutralizing antibodies. Okay. And those neutralizing antibodies, okay, will then do the protection. What is the ultimate aim of a vaccine? Ultimate aim of the vaccine is that it should, when given to a person, it should be able to protect that person from getting new infection. Okay, that is the purpose of the vaccine. Okay. There is one additional feature as I compare the size of the genome. Very remarkable. Students, watch it what I'm telling you. Okay, HIV genome, 10,000 bases long. Dengue and JE, 10,000 to 11,000. Okay, this COVID RNA is 30,000, 3 times more genes. I wouldn't say genes, the length is 3 times more. Therefore, it encodes for more genes. Okay, it has a very fascinating way of making various RNA that will encode proteins inside the cell. Okay. That is very remarkable. there is additional feature of this virus. One of the proteins, okay, it actually takes care of repairing the mistakes that are usually made in RNA viruses, making from one copy to the other. Here is a virus with very remarkable property that there is a proofreading mechanism. So what is the consequence of such a protein being present inside of this virus? You do not see that much mutations. Okay. And you would appreciate that more than 90% to 90 to 99%, the China sequence, the European sequence, and Thai sequence, French sequences, and Indian sequences, not more than 90% they are saying, not much changes. Okay. So I thought this is very remarkable feature of this virus. Okay. Sometime back, we had published a paper on SARS virus. You can look it up at JVirol. What we found that this virus suppresses an innate immunity. See, virus, okay, students, viruses, when they enter, they have to face a powerful enemy, and that is the cellular machinery. Okay, that is the innate immunity. Okay, but the viruses are also very smart. They slowly and very cleverly come across one barrier after the other. Okay, so there are small RNA. It was one very elegant talk by Deepak Modi. You all have enjoyed. The small RNAs that are made by the cell. Okay, these are called RNAi machinery. Okay, they constitute the RNAi machinery. Okay, so one of the genes, I think 7A, it let me now quote a Nobel laureate. Okay, Joshua Lederberg. What has he said? He had said, the mankind will forever, what's the word? Forever keep fighting with bacteria and viruses and all pathogens. But the war, who will win the war? The war will be won by pathogens, bacteria, viruses. Why? Because they can replicate faster. They can adapt faster. Okay, so these are great words of a Nobel laureate, Joshua Lederberg. Joshua Lederberg. Students, go to the Google and see what has he said so many years back. So as I said, okay, there are now coming more specifically to the vaccine point. Okay, this is the crux of my lecture. Okay, so this is the crux of my lecture. Yeah, so there are many vaccine platforms. Okay, and I'll briefly go over each one of them. Let us take the COVID structure. Okay, COVID virus is any platform that you use. Okay, one thing is common, that you need to have the spike protein. You cannot make vaccine if you do not have the spike protein. So as you can see, you can have a DNA-based vaccine where the DNA with the, as we all know, okay, what is the central dogma? Students, you know all of it, okay, DNA to RNA to protein. So you take the DNA that will encode for this RNA spike protein and then just inject into muscle or something, and then that protein is made inside the cell. Once the protein is made inside the cell, then that will create an immune response, and hopefully that will create a protective immune response. I must say that animal models are very, very key to understanding various aspects of this vaccine. Okay, for example, I think some ferrets or mouse studies are very fundamental in understanding COVID infection and natural cure, people who get naturally cured. So, and what antibodies role can play. So animal models are very crucial. Okay. As I said, DNA platform, I have just explained to you. So if I have to make a DNA vaccine against COVID, of course, there are tricks that not everybody will tell you. So what is the line of research? So you make changes on the DNA structures, you can have some CPG islands and what promoter you choose and what kind of immune response. So usually, and sometimes you have combination, DNA prime and then protein boost, okay? So you do it with DNA that will encode your COVID, and then you go with purified protein. So this is called DNA prime, then DNA followed by protein prime. This is one approach to get. The purpose is when you do the second dose, okay, you should get a secondary antibody. Memory cells should form, and you should get a large peak for this antibody, okay? Now, next, let's take of RNA vaccine, okay? This is relatively new, unexplored, not explored to a very large extent. And most people that you hear today about the Moderna, which in USA is just ready to be launched, okay, and found 95% efficacy. So any vaccine, very briefly, I will tell you that it has to go through many phases, phase 1, phase 2, phase 3. Phase 1 and 2, immunogenicity, tolerability, those are the things that are studied, but phase 3 is the big one, okay, where you have to have thousands and thousands of people, you divide into 2 groups, okay, one will receive your so-called vaccine, the other one will not receive the vaccine, but just plain buffer or what we call placebo, okay, and these are double-blind studies, okay? So students remember, phase 1, phase 2, phase 3. Phase 3 is the efficacy trial, okay? And you need to choose a population that is very prone to infection, okay? 50% people should get infected, okay? So in the vaccinated group, if you show that there is protection, 50% people have not got the infection. In the placebo group, control group, people have got infected, then you can say, oh, this vaccine is effective. Okay. As per today's standard, okay, WHO standard or the CDC standard, if you can get 50% protection, then that can be introduced in the population, okay? So students, very briefly, I've explained to you what is phase 1, phase 2, and phase 3. And India, many of those are between 2 and 3 from Bharat Biotech, and many other candidates are in the pipeline. The Serum Institute in Pune, along with, I think, in Oxford, they are far more advanced. They have done phase 3. I think Moderna has also done phase 3, and they are ready to be launched. Okay. Or then there is another inactivated vaccines. Okay, so what do you do with inactivated? You can take the virus, grow tons and tons of COVID virus. I will grow in my lab. Okay, and then there are well-defined methods to inactivate the virus. You can heat inactivate. There are some chemical treatment you give. So the virus is essentially non-infectious. Okay. but then it has all the immunogenic epitopes, okay? So that is one approach where you can use inactivated vaccines, and there are some success against this inactivated vaccines. Now comes viral vector sequences. Okay, this is where the viral, there are many platforms, okay, although you hear adenovirus based, so what you do, you take adenovirus, okay, and then, as I said, adenovirus can be engineered to express the spike protein, and that is what Serum Institute and Oxford, they have done. And with adenovirus, there is a history. History is very important. The adenovirus has earlier been used for many other vaccines, so we can pretty much assume that this will be safe. Safety is very important. So history is very important. Another vaccine platform which has not been, it's not, I'm surprised it's not in the market, is the pox virus based system, where actually when I was in USA, I worked on this system. And believe me, there are many vaccines that are using this pox virus based system. Again, the history is very important. You make, so see, we were children, we were given this box, you can have this in the signature on your arms, upper arms of the, oh, this is box that has been inoculated those days. So again, how is this engineered? It's, the technology is there that you take a pox virus, it's a DNA virus, okay? Pox virus, and then you can insert that COVID spike gene in it, okay? And that will make tons and tons of this protein. When you will inject, immune response will be made. So that is the viral vector. And there are many other platforms. I do not want to get into all this, all the details, but those are less explored, okay? Then let us say, what is subunit vaccine, okay? You don't need the full virus, okay? So you can play with small parts of the virus. You don't need, some people believe, you don't need the entire spike gene, okay? Maybe small parts of spike gene is enough to give you that protective immune response, okay? So there are some techniques now to make it more immunogenic, okay, by engineering of epitopes. So this is known as subunit vaccine, okay. Lastly, I will touch upon the live attenuated vaccines, okay. See, live viruses, of course, there's always a danger. Live viruses, for example, if you keep on culturing in the lab, okay, this COVID virus, okay, keep on culturing for months together, okay, all of us, and then see, and then you ask a question, has this virus changed over time? And they do change, believe me. So from very virulent, they can become not virulent. You can do those mouse experiments. Sometimes this is very easy to see it on a tissue culture plate, okay? Earlier, the virus was, there's something called plaque formation by virus. Plaque formation is, you take a monolayer of, let's say, a mammalian cell plate, okay, add virus, and then you watch for maybe 4 to 5 days, and you ask a question where the virus has dropped in on that plate, how big is that plaque formed? How many cells it has been able to kill? So earlier, if you see that it was able to kill much more, and as you progress, as you culture them over the time, you see that the virus is now making very small plaques, a very small amount of virus cells are being killed. So this virus has been attenuated, okay? It has all the components of the virus, okay, as earlier, but then because of certain changes in the virus, okay, but it is good enough to give you immune responses. So There are some attenuated vaccines, that's how they are made. So in a nutshell, all that I want to tell you is this. There are many vaccine platforms. All of them should have, and they have, the spike protein, either full protein or the part of it. Part will be subunit vaccine. Whether the immune reaction is protective or not, how long does the antibodies remain in the body? Those are some research questions. Are people equally susceptible to infection? That kind of research is also going on, and we, in this institute, we intend to take and address these questions. So I think I will end my talk here about, this is the vaccine platform, and just some interesting observation from my lab at National Institute of Immunology in New Delhi. With all humility, my lab was the first one to show that Delta 32 of CCR5 is present in India. Okay. And we showed it in 1987. Okay. In 1986, CCR5 was discovered by none other than Ed Berger at NIH, Maryland, USA, when I was doing my postdoc there. So we showed that this individual had Delta 32. We published 2 papers in 1987, and we had to do it quickly. I had just joined National Institute of Immunology because it was a very competitive field, okay, to show. And also, let me give you some very interesting information. Delta 32 in India is extremely rare. Okay, we do not find homozygous individual. Those students who are interested to know all these findings, please type my surname. Okay, I have a surname with SNP, which is Banerjee, JEA, and then space A, and you will have all the papers in PubMed. Okay, so this person was heterozygous. We screened 1 in 30 initially. We found 1 individual heterozygous. We sequenced the gene. 1987, we sequenced the gene, okay, in the lab. Those were the days when we used to run long gels. And now, of course, everything is outsourced. The distribution is very interesting. Okay, those people who have this deletion, homozygous, they are protected against HIV-1 infection. HIV needs 2 proteins on the host surface, CCR5 and CD4. If you do not have CCR5, what we call macrophage tropic virus fails to enter. Now, this is a huge information, okay? This information was published in Nature, Cell and Science in 1986 or 87. Okay. And with all humility, you were the first one to make small RNA, small interfering RNA against CCR5. So this was interesting. There are many interesting stories in the lab. You know, when you work in the lab for so many years, there are many interesting stories. Of course, life of a scientist is frustrating, sometimes very exciting. So this is the cycle. But fortunate that my work was funded very well. Okay. So I would like to end with one cautionary note. And that note is, please note, people get infected with COVID, and then let us say 70 to 80% get natural recovery. And there are some studies to suggest that antibodies may be playing a big role. So those instances where you have natural infection and then recovery, the chances of making a vaccine is bright. Okay, in principle, bright. And I'm hopeful this vaccine would come up very soon. And there are many platforms, maybe 30 to 40 vaccine candidates are actively participating in it. Okay, maybe a dozen or more of them have entered phase 2, some phase 3, and we shall have a market that there will be. With Moderna, as most people see, RNA is very sensitive. RNA is very sensitive to getting cleaved. Okay, it's very difficult. Those who work with RNA will know that it gets cleaved, it gets destroyed in no time. Your fingers have nucleases. Okay, so Moderna RNA vaccines, they have to be stored in ultra zero temperature, for which you need machines. So India, I see, will be a huge challenge, and it will be only cosmetic. The other vaccines that the Pune is making, Astra is making, that will be much more applicable, despite having less efficacy. Okay. There are many other candidates that will come up, and we will see how do they perform. Okay. There are some long-term effects of these vaccines, okay, and those have to be studied, and that will take a long time. But then, given the urgency of the situation, I think once the phase 3 trials are over with no adverse events in the groups, we should be ready to launch. Okay. So with that, I think I would close my talk. Thank you so much for your attention.
Thank you very much, sir. It was really interesting, and we came to know the different types of vaccine. I mean, vaccine to different types, for RNA, DNA, or it is directly a subunit. So it is really interesting that all these vaccines are to different proteins or RNA or DNA. So it was very interesting, and we learned your work also, and hopefully we'll be able to go to your website and have a look at your papers and see your. We are very thankful that you have come here. It was Dr. Subir who suggested your name, and you immediately accepted the invitation to talk here. We are very happy, and we learned a lot from you. And please keep in touch, and we would go back to you sometime later to request you.
Most welcome,
so much.
Thank
you so much. Thank you so much.
Thank you. Thank you all. Yeah.
I request Vidyavardhini Madam to summarize the entire, all the 4 sessions of both the days. Over to Professor Vidyavardhini Madam.
Good evening to one and all the participants, the convener, and all the resource persons. A very good evening once again to all of you. So today it gives us immense pleasure, like as the whole faculty of Department of Botany, for conducting this 2-day workshop by science, which is organized by the science academies, and where we have dealt with the ongoing global crisis, that is COVID-19, basic to clinical. So whatever is happening in the world, which is this pandemic in the world, which has created so much of havoc and which has changed our whole lifestyle. We have been living a different life pre-COVID, and the lifestyle post-COVID is changed. So that's the reason the seminar was organized under the convenership of under Dr. Nanderkar Madam. Thank you so much, Madam. And Tarla Madam, thank you so much.
Thank you very much for organizing it very well.
Thank you, ma'am. It's all by God's grace and blessings and support from all of you, especially you who have done this. So we had a talk on all the aspects of COVID. We had different resource persons on day 1 as well as day 2. We had Dr. Nishi Madam, and we had Dr. Tarla Madam today as the inaugural speakers. We had Deepak Modi sir, and we had Rahul sir. So whoever has done, all of them has spoken about COVID. Dr. Haleem Khan also has given his valuable presentation on the global scenario. So from the day 1 till the day 4 evening session, we have dealt about what COVID is, how COVID is dealt, and how COVID can be overcome, what are the possible vaccines which can come. And especially today, being a woman, it was very nice to listen to some of the most of the talks where we have been shown, rather, even we have been shown a video also by Deepak sir, which was focusing mainly on the woman and how the the COVID affected woman can be can have a good insight on this, and how they can survive under all these circumstances. So we are very glad to have all of you, and we are very happy about all the lectures. And ma'am, especially Tarala ma'am, told about the fertilization process and how it happens and how the woman can be safe, and what are the consequences of the COVID, and how we are able to survive in these conditions, and how we are going to survive post COVID. And I thank you one and all. I thank Dr. Haleem Khan, Dr. Aruna ma'am, and the Honorable Registrar, Telangana University, and last but not the least, Dr. Tarla ma'am, for doing all this. Thank you and God bless.
Dr. Vasudev, principal of our nearby degree college. Dr. Vasudev, can you speak for a while? Dr. Vasudev, your made co-host, Dr. Vasu.
Yes, sir. Yes, sir.
You can switch on your camera and you can give your responses.
Sir, good evening, sir. So I'm Dr. J. Vasudev, principal of Vagdevi Degree College. I have attended the 2 days of both the sessions of yesterday and today, and we learned so much about the COVID and present scenario and the current situation in the global. And I thank to the organizers and the speakers. Thank you very much, sir.
Ms. Shirisha, can you speak out? Your co-host now. She's a scholar come lecturer in our nearby college. Over to you, Shirisha. Shirisha, can you speak? Unmute yourself and speak out few words. Are you there, Shirisha, or not?
Hello, good afternoon, sir.
Yes,
Hello?
you can speak out. Yes, you are audible.
Hello?
You are audible, you can speak now.
Hello?
You are audible, you are hello, don't repeat hello, you just keep on talking. Technical difficulties, we'll ask Rajagala, can you speak? Anyhow, it is an online platform and we can't wait much more for the time. Aruna ma'am, can you summarize the entire two-day program? Anyhow, you have summarized so beautifully the morning session. I request Aruna ma'am to speak overall emphasis the entire proceeding of the 2 days lecture workshop.
Yes, sir. Good evening. And the day has come where we are in a position to conclude the program. So this was a 2-day event, and it's a grand success with all hands on the platform, and it's virtual mode of workshop being conducted, organized by our department. I thank all the resource persons who were with us for 2 days, and I thank our registrar, Professor Naseem Madam, who supported us. And I also thank Science Academies for giving financial assistance, that sanction of assistance to conduct this program. And last not least, audience are also gratefully acknowledged, without whom the program would not be succeeded. And of course, I have told all about the lectures and talks. So yesterday, as it was the first day, we had inaugural program by our registrar madam, and then we had 4 speakers, 2 in the forenoon session and 2 in the afternoon session. It was Nishikanda Nayak madam who spoke about structure of novel coronavirus, SARS and COVID, and then followed by Dr. Yogesh Shoche. And he dealt with diagnostic tests for COVID detection. And it was also our head, Dr. Haleem Khan sir, who very well presented his talk. And it was really delightful. And then it was afternoon session where we had lectures by Vinita Deshmukh madam. And really it was wonderful presentation. And she was from Ayurvedic college actually. And it was a totally different area for us. And today morning, we started our program where we had a lecture talk by our convener, madam, that is Sarala Nandekar, madam. And we are gratefully thankful to you, madam. Your lecture was really wonderful. Already we have told that it was all about female reproductive system. And then we are also thankful to Dr. Deepak Modi, sir. And he's an eminent scientist, where he spoke about COVID-19 in correlation with reproduction, especially in male and female human bodies. And then he also dealt with molecular techniques, CRISPR, which was really interesting. And your animated video was delightful, sir, where we have seen to the integrated structure of formation of this protein, that is Cas9 protein. And then this CRISPR is one of the advanced technique in molecular biology, which is to be followed by almost all the researchers and scholars. So hopefully all the scholars might have been benefited. Thank you very much, sir. And then we had Rahul Krishanji Gajbiye, sir. And he also spoke about the impact of COVID-19 on pregnant women and newborns. And we're also thankful to sir. And next we had a delightful lecture by Akhil C. Banerjee, sir. And he's from Institute of Virology. And he has also given suggestions and advices to our students, where students can approach him or can also visit his website to take references of his published papers. So with this, I'm thankful to all the resource persons. And once again, I congratulate our head, Haleem Khan sir, who is the person behind this program. And thank you once again, Tarala Nandekar madam, for being convener of this program. And hopefully we will be in a position, or we are confident enough to write many more proposals. And if sanctioned, we are ready to organize many more programs. And with this few words, I would like to conclude. And thank you, sir. Thank you for giving me this chance. Thank you.
Now I request Tarala madam to give her remarks on the overall 2 days lectures. Tarala madam, over to you.
Thank you very much. I think the way you have organized is wonderful. I never expected things will turn out so well, because this is online and it's first time I'm attending an online program. And maybe you are experienced, but I have done it for the first time. But it was really grand, and the continuation and the topics, everything was very good. I think all the speakers were so good that I don't know whom to praise more. It's really, and we know the basic and clinical also aspect. So I think the title goes very well. what we thought of is very true. That is from basic to clinical, we have gone ahead to understand the COVID-19 pandemic. You have organized and your people have worked so well that I don't know how I should thank. Of course, I have to thank the Science Academy who have supported us. And can you give us the YouTube address, how people can go to the YouTube, which program it should be? I mean, that if you can give the title of that program, it will be easy for other students to have a look at it. Thank you very much. It was well organized, and I congratulate all of you, not just you, but your team. It's wonderful. Thank you so much.
Yeah, it is a proud moment for us to explore and have a vote of thanks. But I express my deep sense of gratitude towards all the resource persons, and special thanks from my heart, bottom of heart, to Dr. Tarala Madam for her acceptance. Soon the communication was approached to Ma'am. She was very, very supportive and encouraging to move forward. And when we started the initiative, we had a limited number of resource persons, but soon the days progressed. We have roped too many speakers, too many people from Kerala to Mumbai. So almost we have traveled west to south, all speakers we had, just because of her initiative and her interest to make young generation, or we young teachers, to it is a long way to learn more and more from you. Though it is a virtual mode, the feeling of attending a great charismatic people or the stalwarts of the science by sitting at home, it was a great amusement and encouragement from the blessings of elderly people. So with these few words, I would like to thank Dr. Nishikanta Naik ma'am from Hafkin Institute. I would like to thank National Science Center Pune, Dr. Yogesh sir, and Dr. Vinita Deshmukh ma'am from Ayurveda Mahavidyalaya, Wagholi, Pune. I would like to specially mention thanks to Deepak and Modi sir for enlightening the two lectures in one with one break. And I equally express my deep sense of thanks to Dr. Rahul sir for giving such a informative and entire whatever the institute today has done. The institute has given a very clear concept, very clear clarity on reproductive system, and also it has given insights on entire reproduction and how the corona is doing in all this duration was beautifully made, like Zika virus, MERS virus, SARS virus. Dr. Rahul sir has given a very clear insight, and Dr. Deepak sir has given a very clear insight on the male reproductive system and female reproductive system, and even pregnant women. What are the receptors of this virus was very clearly told. And the initiative of today that Nandedkar madam has given very clear how the pregnancy, how conceiving happens among all the young students, participants who are joining this. And it is a great time to have a stalwart director, Institute of Advanced Virology, Trivandrum, Kerala. Out of his busy schedule, he gave us time to get focus on the vaccines, and in the corona vaccine, how it is going on, what are the approaches going on. He gave a clear-cut insight, and it is time to thank the entire cast and crew of panel of Science Academies. I express my deep sense of gratitude towards Professor M.R. and Murthy sir, chairperson for the Indian Academy of Sciences. And also I express my equal thanks to Dr. C.S. Ravikumar sir, coordinator of this lecture workshop and refresher courses. And a few words about the academy is, academy has been started long back. Indian Academy of Science Bengaluru was started in 1934 by Sir C.V. Raman sir And in 1994, along with University Grant Commission, the Academy has focused to perform many science promoting activities in and around the country. So in 2007, all the three academies, Academy from Prayagraj, New Delhi and Bengaluru, has joined to make a joint panel where from 2007, three activities have been started by the academies, that is summer research fellowship, refresher course and lecture workshop. Summer research fellowship is during the summer for the students of pursuing masters and research scholars and teachers of the young and new career. And also refresher courses offered, already Tarla madam was a convener for number of refresher courses, and also she has equally contributed for convening the lecture workshops. Till date, on the website of academies, Indian Academies Bangalore, offline and virtual mode workshops are 1,115 supported by the three academies. With these words, I equally thank the entire Department of Botany, the university authorities, and publicity done by Mr. Naveen, computer. He is a computer expert supporter. Mr. Naveen has made wide publicity by posting our flyer on the university website, and I equally thank our department members like Dr. Devraj Srinivas, who has covered in daily news today's at least more than 5 daily newspapers has covered our workshop, and it was in regional language, and more the today's dailies have covered Nizamabad special edition. They have covered more focused on Ayurveda as an immune booster. The headline was given as Ayurveda as an immune booster, and it was almost in the district and almost in the state. It was widely circulated about our workshop. Before the conduction, pre-workshop and post-workshop also we have covered in media. And we equally thank to Dr. Jalandhar Wagmare for contributing his write-up for newspaper. And we also equally thankful to Dr. Triveni, the PRO of the Telangana University, who is keen in focusing the activities conducted by the department to the regional local newspapers. Dr. Triveni and PRO team is also equally thankful, and I also equally express my deep sense of gratitude towards my two sisters, Professor B. Vidyavardhini and Professor Aruna ma'am, for their constant blessings and support in and around my activities. So I equally thank the scholars who are actively participating in these 2 days event. Without this participation of the scholars, and scholars are also active teachers in various institutes, and they are also promoting. They have promoted a lot among the student community about this event and meeting, and it is equally thankful to the entire people who have very cooperative, and the feedback link for the certificates has been posted in the WhatsApp YouTube link, and shortly we have created the WhatsApp group, 2 groups, where we have got more than 534 participants registered, enrolled, and with a few people, 13 or 14 people whose email IDs were wrongly spelled in the in the registration form. Kindly, if you submit the exact email IDs, we will be trying to share the YouTube live streaming links to the all 534 people who have registered for this virtual mode workshop. So it was we have taken permission from the academies for only 100 people, and we have gone 534. So it is all it is all the glance or overview of the Madam Tarla Madam, who was able to make this workshop resource persons from different nooks and corners of the science field. So by looking the list of speakers, people have keep on joining on to the workshop registration form, and we have crossed more than 500 participants. With these few words, I would like to take permission from Madam to end the meeting. Over to you, ma'am.
Oh, it's wonderful that 500 people have come. Of course, the limit was not there because we didn't have to go give lunch to them. Otherwise, they had to restrict it to 100 only. Earlier, they used to restrict it to 100 or maximum 150. So here it's open online, so people can go directly and have a look. So can you send the link, the YouTube link, to me also and to others, so that they can go through and give the response also. And maybe if you have a response,
yes,
the responses you have received, those also can be sent to the academy. They will also like to know the response to these lectures. Okay. Thank you very much to all of you who have supported him, and you yourself have worked on it. So thank you very much. That's all. I think the speakers, Science Academy, and you, the group, together, we should thank you all. Thank you. Thank you very much.
Thank you, ma'am. Both Vidya ma'am, Arun ma'am, we are ending the meeting.
Yeah, thank you, sir.
Thank you, ma'am. Thank you, everybody.
Thank you. Thank you.
Thank you, everybody.
Thank you, everyone. Bye-bye.
Bye. Bye, sir.
Bye, madam. Thank you.